All right, why don't we get started? Welcome, everybody. Happy New Year. Today's webinar is going to be with Elucidata, Curating Biomedical Molecular Data. Today's agenda will be a speaker introduction, a brief industry perspective, Elucidata presentation and demo, and then a Q&A to follow. We also will have a poll, two polls before we hand it over to Elucidata. So about Elucidata. Elucidata's cloud-based platform Poly delivers machine learning ready biomedical molecular data that helps in standardizing, streamlining workflows and analysis for biomedical experts. Hosting a rich repository of more than 1 million plus biomedical data sets, Poly uses state-of-the-art proprietary AI-enabled technology to assist in transforming data so that it's analysis-ready and is used to discover assets in oncology, immunology, and other diseases. Today's speaker is Dr. Richa Mughal. Richa is a senior bioinformatics scientist at Elucidata with more than 10 years experience developing algorithms and generating insights for drug discovery. At Elucidata, she's involved in understanding customer data curation requirements and translating them into scientific and technical plans that help customers achieve their research objectives. She also helps in building scalable and machine learning-based solutions for public and proprietary data curation, custom automated workflows, and data analysis and visualization on their cloud platform. So today's rules of engagement: put your questions and or comments in the chat or Q&A box down at the bottom. General questions will get moderated after the talk. Be respectful and let's have a really nice Q&A. It's a great opportunity to ask and get involved in the webinar. And then afterwards, you all have access because this will be recorded. So I'm going to go over, this is John Conway, I'm going to go over a couple industry perspectives, maybe some definitions around data science, data curation, and then we'll do the poll, and then Richa will take it from there. So I know a lot of people have heard these already, but it's always good if somebody's on here who hasn't. What is FAIR data and process principles? It means findable, accessible, interoperable, reusable. That's what we're striving for in our data, scientific informatics and data environments. It's not just about the data, it's about the data and the processes. What we're trying to achieve is model quality data. You want high data integrity, you want it to be highly contextualized, you want it to be assessed with confidence, and you want it to be applicable so that you know exactly how and when you're going to use it. And I will say that we are on the cusp of a lot of new things coming in scientific informatics. It's in my opinion that it's in its infancy. And we are working closely with a lot of companies like Elucidata to help change that. Data science. Data science is critical because without the data science today, we would not be getting the insights that we are getting. So data science is where it all comes together from a computer science perspective, a maths and statistics, the machine learning, the artificial intelligence, and then really having that domain or business expertise that allows you to get those insights that you need, convert that data into information and knowledge and intelligence eventually. And this is really critical for what we're trying to do today because it's highly complex. And then data curation. Data curation goes from data strategy into data assessments, contextualization, annotation, extraction, ingestion, and ultimately into a fair data environment where people can reshare, share, and take full advantage of that highly curated data. And that's what you're going to hear from Dr. Richa today. So with that, I think we're going to run two polls. and then we'll hand it over to Dr. Mudgao. Aaron, are you able to run those polls? Yeah, there you go. So as you can see, the question is, what's the volume of data you generate in a year? Most people are in the terabytes region. You're depending on who the folks from what they're doing, petabytes. As you get into multiomics, and companies as a whole, large companies will start to produce exabytes of data. All right, why don't we end that poll, and then I'll share the results so people can see. Oh, I'm sorry. Aaron's supposed to be doing it. So you can see that for the most part, between gigabytes to petabytes is where the normal range that people are in today. Sorry, Aaron, didn't mean to interrupt you there. All right, poll question number 2. What type of data do you commonly work with for your biomedical research? So the horse out in front is Multiomics, which is great to see, with a close clinical and mutational studies, and then imaging data, and lastly, EHR. Looks pretty stable. And so you can see 82% is multiomics data, 21% mutation studies, 29% clinical data, 16% imaging data, and 3% EHR. So pretty cool. All right. So now we can hand it over to Dr. Richa Mudgao, and she is going to give a great talk and demo. And we'll stop.
Right. I'll just start sharing my screen. So can everybody see my screen?
Yep. Thank you.
Perfect. Right. So let's just get started. First of all, thank you everybody for joining us, and thank you, John, for that lovely introduction, and also setting the stage of what the biomedical data is, the current status, plus also what verification is required, and the data is stemming predominantly being unfair in nature. So what I want to do today is, in this very quickly referring to what's the agenda for today's webinar, is that we would like to first walk you through what are the current or inherent challenges that stay with us in the biomedical data space. And a large portion of my of the webinar, I would like to take you through a case study for a real life customer that we had, and the questions they had in mind, and what were the challenges that they faced to go through them, and how we help bridge them the gaps that were predominantly arising due to the data being not usable for them, and curating them on the go for this. And finally, just to give you a brief of like what we do at Elucidata, and how did our curation solutions were able to help them in analyzing the data and moving through their pain points. Right. So I think before we, there is a huge amount of data. This is something like in the past 2 decades, we know that large amount of data gets generated. There are high throughput experiments, and like as we can see from the audience itself that's attending today, a lot of them are related to multiomics data technologies, and we have seen a boom in such texts, and then a lot of data that gets generated. Another thing that we're seeing over time is that there are very wearable implants and those kind of devices, which could lead to a lot of data that gets generated. What is common amongst them is that although the data is getting generated, that it's largely unstructured or maybe in a semi-structured form, which kind of hampers you to extract out the real opportunity that really lies in this data. So what we when we talk to our customers, they want to, most of their discovery projects or the biomedical research, one common thing is that they are very data driven or data heavy, and they would have different sort of questions which would be related to, maybe the structuring of the data itself, or to source the right data, or for the lack of an infrastructure of the data. It will just give you a flavor of what different, what we really see in the real world or the kind of biomedical research, is that there is maybe there are a lot of clinical data that's getting extracted from publication itself, or they run custom bioassays which are or there's legacy information that's there, and this data in itself, when they are sourcing, when they lacks a structure and lacks metadata annotation for them to really extract out the information from there itself. Another important aspect is that they want to probably procure data from outside sources, which and want to validate their own hypothesis, but they would lack expertise in first in sourcing that data, or uh, first finding that available data itself and then making it usable for their research. Another thing, like we also wanted to understand from you in the audience, like how much of data that you're generating, and with the boom of new technologies, the data generated itself is huge. So processing it and storing it itself, it takes a lot of infrastructure requirement that's there. And many of times when we, when teams are growing, or when we have when we have new early stage companies, and they keep generating more work, and when you're expanding their teams, they are often, the data gets scattered, that it takes a little it's difficult for them to organize it also, and then to collaborate itself. So a lot of their teams then would start working in silos, and kind of hinders their collaborative technique there. So as even John mentioned, main problem that we see is that the data at source is not always in the most usable form. And when we say that it's not usable, what is the main that the different hurdles that one has to overcome to really start understanding what the data is available and how can they extract value from it. Many a times when you have the data, it's even in the most known, sources such as Geo, there is only like less than 3% of the data which is actually curated or you annotated with metadata. The lack of annotations or metadata itself would hinder you to reuse them or to actually find your data in itself. The other thing that one is that the amount in the formats they are available, they are not always in a very consistent format. They're not always in one standard way of processed, so which hinders or reduces the interoperability of these data sets, and also reusing these data sets. And as we said, the unfair practice is also about in the existing sources is here hampers the accessibility, and the lack of infrastructure also hampers how do you process them or store them at scale. So at what at Elucidata we understand is that because of these hurdles, we have a fair bit of understanding of like how to overcome these hurdles and how to break these barriers to make the data which is available at the source in a curated format for you to extract value. When we talk about data at source, here the data at source could be, say, public resources as well, but it could also be the legacy information data that's lying within the company or the organizations. At your And that itself may not be in the most usable form for, say, others in your organization. So what I would like to do now in next few minutes is to walk you through a real life scenario of a customer who came to us with where it was an early stage company, and they wanted to leverage the information within their proprietary data set, as well as data sets which are available in the public sources, to identify biomarkers for their women's health disorders. So they're very passionate about disorders related to women's health issues, and then they wanted to make they were predominantly working in the area of microRNA and transcriptomics, and they since they're an early stage company, they did not have, right now, the infrastructure to run large scale or experiments, and also not to run single cell experiments. So they just want what they came to us with were like three separate problems. One is that could they use the microRNA information and integrate that with the transcriptomics information that they're generating. Can be they were working in two different teams, and very specifically they had the two different teams follow their own ways of, vocabulary of how to name their tissues and samples. So at some stage they were struggling to, understand how to combine them. And at the same time, they were all, since microRNA is known to have, they wanted is known to affect different menstrual cycles. They wanted to also understand if there is available single cell data that could help them in, integrating. So could they find public studies to in the single cell domain? Another thing that they wanted to do was that since they're running microRNA, they also wanted to see if there is something already known, and how would they really claim that the research that they're doing or identify something which is novel and not available? And this is a key problem that many people face, that once they find something, and in order to claim that that's truly novel, they really have to dig deep into the public resources and identify whether that research is really novel or something that they were really not, it's already known and validates in some ways or sort. And a third key point is that since they were a early stage company, their collaboration across a centralized place for them to access all of the data together was a challenge in itself. And also the infrastructure to process data, with a cloud platform or so. So so what I would try to do now is what they wanted to do, and in doing so, we help them identify what's really the key pain point for their analysis, and then help them overcome those key pain points. So coming to the first thing that they wanted to do was they had in the in-house generated microRNA and transcriptomics and mutational information that they had created, they had generated, and they wanted to leverage the information that's contained within all these data sets to identify some biomarker part. But when we spoke to them, and when we tried to understand what their data is, we clearly identified a lot of key problems, mainly because how were they really there were inconsistencies in how they were storing their metadata in itself. So the transcriptomics group had a better way of storing their information of the samples in the metadata, say in an Excel file, but the other group did not follow the same, and so for them to integrate became a problem in itself. And also there were challenges in terms of, because they did not use a control vocabulary to name the samples, and how do you identify the different assays they were doing. And also another thing is that whenever there is manual curation or manual part involved, there are discrepancies, there is incompleteness. They did not often fill up all the things, and sometimes there were few fields which were missing. So the incompleteness of the data also posed a great a challenge for them to start using this data that they're generating and integrating them together. So what we do, what we understood after understanding the different pain points, we have a curation workflow itself. And although it's a workflow, it generally has to be customized for the customers as such. But we have broken down at least the main in data ingestion to understanding your data and understanding what you require from your data and how you want to use it, followed by harmonization of the metadata and putting them into a real standard schema or a structured format, and processing it through a standard pipeline so that it improves interoperability and combining. And finally, the consumption assay. So we have a framework of sorts, which is the curation workflow, and then we customized this based on what we understand from the customer and the data that they are providing to us, which could be proprietary source. It could be something that they derive from different publication. It could be some of the internal biochemical assays that they run, or it could also be some of the public sources that they are interested in, and they want some. They know these are the list of data sets that they want us to, make it usable for them. And in short, also often it takes a huge there's time to process your data from source to really start making insights again. It takes it's a long process, and sometimes you may lack the expertise to even do such things. So this is I would say one of the favorite steps when I do any new curation related project. This step is very, very critical, where we try to understand from the customer what the data is really telling us about. The first and foremost step is that when we get a curation project, we try to gather the team of experts that would have enough subject area knowledge and could help them curate their data as well. We try to understand what is the source of the data, and how was it shared with them, or how they could share with us. We also want to identify who will be the users of the curated data. So even though standardization and usability is an issue, it's also important to understand who would be the real users and how would they like to use. And also it's important for us to understand how would they use the curated data. If is it for integrated purposes or multiomics analysis, how would that essentially help them in using the data as well. Once we understand that, there is a large chunk of time that we like to understand from like what are the processing needs, understand what define the standard schema, because the standard schema in itself for different data sets would use the enhance your interoperability and understanding and using the data. Another important thing is to understand the metadata fields that they have generated. So whenever they're running custom bioassays, they would define different specific tissue types, which would be more important for them. In maybe when you are looking at different data sets from the same in the public domain, the fields that are curated are more broad and more on more umbrella terms, whereas they would have gone at a much granular level of defining these, and this is something that they would want to attach with their data to make it and to analyze it better in downstream. Another thing that we at this stage we understand for them is one, to understand which metadata fields are important, which would help them identify the data, which would help them integrate the data. And also the metadata fields, when we understand these are the ones that they need, we define what are the ontologies that they could use and what is the control vocabulary that they could follow. At this stage, then we define the guidelines as well. That given a metadata field, these are the different ontologies that they should follow, and these are the terms that they would be using to curate the data. And finally, we derive from them that what are the requirements of the data storage and how would they want to finally consume the data. Would they be more comfortable with a code-based search, or would they also like to do with some GUI-based research and so on. So one of the key aspect, like I mentioned here, is that we spend a lot of time in custom metadata curation, and all of this is, I mean, from the looks of from what we understand, is a very time-consuming step, and but it could actually be automated, at least in semi-automated form. And what we have developed at Elucidata is a curation infrastructure. The infrastructure actually comprises of two parts. One is that the data that we source, the uncurated data, when we define these rules and we define these metadata fields that we need to curate, we add them. We run our own polybot models, which are pre-trained on, these are natural language processing models, which are pre-trained on biomedical data, which helps you annotate or curate the metadata fields as well. And this provides our internal experts, curators, with a weekly annotated metadata already. And over this, once they have that, they are able to correct them if they are not, and that goes back as a feedback loop, as you can see, to the model. And this is the active learning process. So the next time, from a weekly annotated model, over time it improves in itself, and it becomes a very highly accurate model itself. And this infrastructure for us has enabled us to do this, a much more faster, at least 4 times faster than a manual curation workflow. I would like to now just give you a glimpse of how this infrastructure looks like in yeah, just make this a full screen. Is the video playing for you?
Richa, you might want to hit the arrow down on the left. There you
go. The video is not playing for everybody. I'm sorry, I'm not able to see it. Yeah, it started playing. It's ABC. Right. So this is a data set that say I mean, for the purpose of this demonstration, I've got from Geo. So when we ingest a data set from any source, which could be your data as well as it could be a data from a public source, we define the metadata fields that we're most interested in, and these come, and then we define the curation model in the backend. And these get automatically labeled with the ontologies that we would need. The model first identifies these keywords, and then it actually then follows the ontology. But this infrastructure gives us the provision to say correct a different the metadata field. So say if something is already, if it's correct, we just go ahead with it. When something is incorrect, we can just rename. We can add the metadata field into the other, and then mark it as complete. What happens in the backend is not just that these annotations get corrected, but also that the polybot models which was used to it also get retrained and improves with time in itself. Right. Right. So moving from this the polycuration infrastructure that we have. Another key step that we follow is identifying is putting the data in a standard schema format. And for the case at hand, the data that we received from this from the customers was that it was from for different data types. It was transcriptomics data. It was also, they also had microRNA information, and they also had mutation information. And this data that we derived was from different sources that was either shared with us on an S3 bucket in a raw FASTQ format, or it was in a CSV format in just some few folders and drives. And also important to note is that the metadata associated with each of the study, which defines what are the samples used, what are the conditions used, as you can see in the figure below, the different treatment or tissue used. that was also separate from the data and in another Excel file. So this itself would lead to a lot of problems that one would when they want to integrate. It takes a lot of back and forth, or the collaboration between teams becomes even more cumbersome for them to understand where the actual data is, and then where the metadata is, and how does that apply to them. And also the format in which they're available, they may not be processed through standard pipelines, and they were just raw files sitting on their S3 buckets. And when we curated the data set for them, it's before we process the data through a standard pipeline, and also store them in a standard format, which is a DCD format for these. And also we attach the metadata that's available to each of the data sets itself. When we store these data, we kind of index these data sets, which makes thus the queryable part of these data in a much effective manner. Moving to the next thing that they wanted to do was they also wanted to understand from other public studies that are available that could help them in generating, validate their respect. So one key aspect that we need to understand here is that they were running some microRNA experiments, and these microRNAs are generally, very tissue specific, and they are known to repress the gene functions, and they tend to be they tend to be good biomarkers for early detection and diagnosis. They also observed that they had seen one study which was pretty old, but which had about just few microRNA which would differentially express between two conditions, and then they wanted to see if there are any more new available ones. Since they also understood from their requirement is that sometimes the menstrual cycle in itself, there's different stages in itself, could lead to different changes in the expression of the microRNA. Since microRNA are more upstream and they attack they are controlling or the transcribing the mRNA information, they wanted to integrate both the information and understand from the upstream, from the repression part, plus from the genes which are actually changing in the transcriptomic study, and see if they could derive something which is novel and pure. But what they were lacking is that they had to search for these data sets in, so the first start would be to Google these data sets or then look at geos, which itself was posing problems. And they also lacked the expertise to even source these data or also process these data from different data. And mostly we know that most of the data sources are being the databases that are available may not follow fair data practices, and that's predominantly because the rate at which they're growing and the data being deposited by different researchers, it's very difficult to ensure that they all follow standard format or they all upload enough information and so on. So for them, this was also becoming a very cumbersome thing to look at, and if they were to do it themselves, it would take them a much larger time, and they also lacked expertise. So what we have also done at Elucidata is that we have curated data from various sources for predominantly for multiomics technology, which could be related to from links data sets or from TCGA and from CPTA. They would be related to, say, methylation or metabolomics or even proteomics and microRNA data as well. So because we have a curation infrastructure in place, we are able to ingest and process and bring this data, curate this data on a large scale, and then create a database or a Multiomics atlas for like more than 1 million data sets to date. And this, by the sheer amount of data, the volume of data that we're being able to curate, we inherently just improving the search probability of finding your relevant data sets, and and also the time that it takes for you to then get the data set and start using it. It has a very minimal latency, which would take you days, and which could in this case, if you find the data in Multiomics atlas, you can start working on it from the next day itself. The other thing that we have learned over time is that once we have source data from so many different multiomics related things, we now have we now have expertise as well as the custom curation infra that could help us do this at scale. So even if it's a new data source, but it's one of the data types that we know about, it becomes it is for us a much faster journey to take care of. So given the problem at hand for our customer here who wanted to find, we searched in this Polyomics Atlas and we found data sets. They were predominantly interested in two different types of information. One was for endometriosis and one for breast cancers. And then we helped them procure these data from the Polyomics Atlas, mainly in the field of transcriptomics bulk, as well as in single cell data sets that we could. Uh, Moving to the third thing that they wanted to do is that once they have collected all of this information, they want to have a centralized place, a centralized system where they could keep, they could collate all of this information. And this would then facilitate them to collaborate between the teams. And also they have a system where they can, start exploring the data and processing the data. So but they did not. They lack such infrastructure, as they said. This is where we created something that we call Enterprise Atlas. And what Enterprise Atlas predominantly mainly does is that it will curate your own. It will curate your proprietary data sets through the polycuration infrastructure that we have developed, which is powered by Polybird, and also populate that with the relevant data sets that we could procure from our own omics atlas. And in this case, there were 16 data sets from their proprietary source, and then we augmented them with about 160 data sets or more further than that. The enterprise omics atlas does not even provide you a centralized space to just store your data and keep the curated data with the GCT with the standard format and the metadata. It's not just a place to store, but also helps you analyze it on the Poly platform itself, which could be a GUI based, which could also be, if you're code savvy, it could also be through our Poly Python notebooks, which is something I will demo in a few minutes. And it also, because through the Poly Python libraries that we have developed, you can also then, it takes preferred to use your own computational infra. You can download or access these data sets at your own systems or your own organizational infrastructure. So I would now shift gear and just take you through the demo of the Poly platform itself. So this is the Poly platform that we have developed at Elucidata. Largely, it has the works. We help you organize your data into workspaces. We have built web apps as well to work on your data set, and then the data that we curate for you, it gets stored into an omics atlas format. And also the data that we curate from public sources gets calculated into an omics atlas here. For the organization here, we had generated an enterprise omics atlas, which the data sets that I just mentioned, we had created about 172 data sets. When you start exploring the data set, the first thing that comes to everybody's mind is that what is my atlas what is can I get a summary of it? So what you can see is that we had a few data sets that we curated for them, and we procured a lot more than 100 data sets from GeoOmics Atlas for them. And this is the data that is available for them to now start working with. These data come from very different source types. It could be transcriptomics data, or the mutational data, or the single cell data, or microRNA data that we have created. We also are able to understand a bit, like a large set of the data that we were able to do were coming from breast cancer data sets that are available in the public domain, which is because we do have more studies related to that, and also a snapshot of what different organisms these data sets are coming from. So so this is a summary, and then let's just view all the data to understand how the data is organized, and once we have a data in such a platform, what does it mean for us? So firstly, we have whatever metadata that's get curated gets added as a filter in the omics atlas. So this provides you a graphical interface at which we could just click and subset your data based on these metadata labels that we'll add. So every data that we have curated would get annotated by these different metadata fields for the different tags. And this, makes the searchability within your Omics Atlas even more faster and easier. Um, so if you can just go by the sources of the geo, we have about 156 which are matching this criteria. They were largely interested in endometriosis, so you can subset them based on that as well. Um, a key thing that I also would like to show here is that these different data sets, as they appear as different, the cards of it, we have the provision to just have to understand or view the details of what the data set actually was about. So when you go to the actual source of it, you will have to read through a large slew of paragraphs to understand what were the actual samples that were used, what was this a treatment or whatnot. That so a lot of that information is actually hidden in text, which in an OMIS atlas like this get very much easier for you to consume. So this data had two different types of cohorts, which were like normal versus patients suffering from endometriosis, and then they were also treated samples versus if they were treated with some drug or not, and which cell type would they borrow, which genetic modifications do they borrow. The data that we come from different sources, we have about 14 different metadata fields, which are very important, which we have defined, and we attach that with every data set that we have, which enables us to compare data sets across different sources as well. Within this, you can also have the process data. Just this is a visualization. So when it comes to the analysis part of it, again, there are multitude of options for this. If you want to, you can simply download the data set, which would be which is the standard format of a GCT combined with the metadata. And if you prefer to analyze the data set, again, we have different examples here at place. Within for this, since this is a transcriptomics data, you can then open this into a PolyPython notebook, which gives you a notebook code base. Or you could also open it into an application that we have developed for you to explore these. For the interest of time, I've already opened them in the sites here. Right. Another thing that I wanted to remind was that they were interested in single cell data a lot, and especially related to endometriosis. And one of the key things is they wanted to see if there are any genes which are within the normal cells. What is the transcriptomics profile? And the data set that they had they wanted to explore uh, was for normal cells for a single cell. And I'll just, so given the different data types, it's easier. We can just, explore them, just sort them out, just get there 26 of them matching. And any of them which is actually related to endometriosis is something that which would appear here for you to, explore further. Um, when we want to analyze this, we are also, it's exploring these data sets again. It could be in the notebook, but also the apps that we develop. But we also have the provision to bring in third-party apps. One of Cell by Gene is one of the most widely used tool to explore single cell data sets. And when we want to explore these, if you can imagine a journey with this data set being available on Geo, versus now here where you could just simply select the data set and open it in, launch the app, and it will then open the data for you to start exploring. And how easy it has made the curation of the metadata has made it so easy for you to explore, where you have the cell type identification, or also the phases in which the different endometriosis cells are there for you to go ahead with to further analyze. Right. Uh, Going back to one of the key questions that we started off with, where they wanted to combine information from microRNA as well as transcriptomics data. It's something that they could easily do it in a notebook environment. And here I have what we have done is that you can it's a code friendly thing, then you can import your data into a instance of your choice. You can then download the different the data set that's of interest. You could also explore or query from other omics atlases that are present, and then combine them into into a final integrative stage. So we were able to provide all of the data sets that they were really interested to explore, and then get into and define the biomarkers that we did. So just going back to the slide, so summarizing that what they were able to do was that from their own proprietary data set, they had a list of microRNA biomarkers that they could find from additional data sets from that we had found for them. They were also able to get a list of biomarkers for that. And they also were able to use some of the predicted gene. So it was a transcriptomic genes and they found the upregulated, downregulated genes from there, where they could map them to they could predict the mRNAs that could there and get a list, another microRNA. And from a combined results of these, they were able to identify some high confident and even a few of them which were novel biomarkers for endometriosis in their study. Right. So finally, I just want to just walk you through why Elucidata is something that you could choose. Is that we have we understand the problems that are there in multiomics analysis. We can say we have taken 4 strong pillars that we can define for curation. One is that we have a strong team of expert curators who have the domain knowledge, which would help you understand your metadata better, help you understand what fields are important, help you understand what are the best possible ontologies and vocabularies to use for your data. We also follow best practices in the curation, which would be domain specific. Given the polycuration infrastructure that we have and the polybot model that we have developed with the active learning process, which is improving the accuracy of day by day, that's something that enables us to do curation for your data or from public sources at scale. And then finally, the accessibility of these data for you to start integrating and playing around through in a GUI or through a code-based platform is made easy by us. And just want to also leave you with the thought that it's not just that we are able to do the curation at scale, is that the speed is the only thing that's important. We also very heavily understand that the accuracy is very important for us. And then we have run a few benchmark studies where we have benchmarked our models against the gold standard of metadata curated by a group of experts. In what we observe is that the models that we have generated for different metadata fields have very high accuracy, and that helps us grow data sets. Something that would take much slower for others to curate. It takes us, we're able to do that at a much 10 times faster stage. Right. At that, finally, the key take-home message that I would like to leave you with is that we try to make your data useful for the whole organization, regardless of who has generated it. We enable you to integrate and perform multi-omics analysis. We also rapidly source data from different different public resources and things, which also this would, because of the largest on mostly are interested in the latest data sets, and you would be able to find that in Polyomics Atlas at much rapidly and be able to start working on it at a much faster speed, so accelerating your research. And finally, is that the data that we help you manage the data and help you consume the data or extract insight from the data in a very code in a environment which could also which is also code friendly and also friendly for bedside test, so on. And with that, I would like to thank you all, and if you have any questions.
Thanks, Richa. Yeah, we have 2 so far. How does one know when the data is incomplete? That would be the first one.
Right. So the data being incomplete, I would say that there are two things that whenever we are looking at data, we would first say data and then the metadata that describes the data. And both of these have the possibility of being incomplete. When we talk about metadata being incomplete, then it's about when uh, we have different fields which are important. Sometimes if the absence of a metadata does not necessarily mean that it's not important and it's not relevant, but sometimes that's what also entails of being incomplete. And within the data itself, there would be that there is a possibility that there are some features in the omics where they have not been captured, and there would be NA values or NANDs that exist. And most often or not, we try to first interpolate that information from the other data that's present. Or if these are incomplete and cannot be extrapolated, then they need to be removed, or the data needs to be cleaned and remove these, so it doesn't have discrepancies further.
Okay, great. The next question: if the metadata of the study is partially provided by a data submitter, what is the best way to utilize such publicly available data sets?
Let me just read through it once. If the metadata of the study. Right. Yeah, I think a lot would depend on the submitter and how and what they want, the information that they have put in. Sometimes I feel that some of the metadata can be derived from other metadata as well that's available. And many a times we have found information, so it may not be part of when they're submitting to CAG, or they may not have put it in the actual tag, or overall experimentation may be empty. But when you actually read through this abstract and you read through the some other text that they have submitted, it could be extrapolated from that. And when we do the curation, and what we ensure is that we define the first metadata fields, and whichever metadata fields that we have defined, we ensure that these fields are filled with the accurate information. Part of it would be, say, derived, part of it would be we would fill it manually.
Okay. Yeah, I think this is part of the challenge of curation, right? When you partial data, understanding incompleteness, and even the hidden parts of it where, we have a reproducibility problem in science, right? So, taking and doing the absolute best that you can with what is out there is the goal. And then the outcomes depend on a lot of those things. Okay. Another question is, do you put any curated compilations back into the public resources?
No, we don't put it back to the public resources, but we definitely put them in Multiomics Atlas. So any data that's curated by us gets into the Multiomics Atlas, and it's available for people to use.
Right. Right. I mean, it's an interesting question, right? Obviously, if somebody responsible for a public source or some of these sources wanted to enhance their data sets, they could use Elucidata to do that, right? So, and obviously there's all different types of methods and stuff, but it's definitely an option. Okay, thanks for the question so far. The last one we have right now, I wish to understand, since your pre-processing pipelines are not open source, and if someone wants to reproduce same results, how do they do it?
Right. I don't know exactly how.
Yeah, it's kind of a question where that's the difference between open source and a commercial source, right? If you guys wanted to make it converted into some sort of universally usable open source tool, then that's probably how it would be. It would have to be something like that. Yeah, I think.
Yeah, but what we would ensure is one thing, is that the data that we are processing for you, as well as the data that we're processing from the public sources, And if they are the same data type or data types, then we would follow the same processing pipeline,
Right.
ensuring interoperability, ensuring that the same standards have been used, the same methods have been used for you to then, in future compare. Or also, like, if you want to, put in some samples from two different sources, well, is that even possible? If it's processed through the same pipeline, yes, it would be easier and possible for us to do.
Okay.
Mm-hmm.
Let's see. I just want to make sure I didn't miss any questions. I think that's what we have so far. Any last questions? We're at the top of the hour. I think that the stats that you showed about the efficiency improvements and is pretty interesting. And I think what you get with a commercial offering is, highly curated, highly reliable, highly supportable. Those are the types of things. And there are a lot of people at Elucidata that are working on these to get these data sets to the point that they're at. It's not like one person's working on them. You're talking about teams of people, right?
So as we understand, like, curation is dependent on what the data is about, and it's usually customized. It needs to be customized for the data that we're in. We do understand what are the parts which could be automated. We do understand, and those are the things that we have, built internally as a part of the infrastructure that would help us do that at scale.
Right. Then we have one last question. So when you come across sources that are so bad as to be liability there, what do you do? And I know, like, we run into this from a data assessment perspective. Like, we'll assess the data environment to say it's good, bad, or ugly, right? But go ahead. It'd be interesting to see how you guys approach it as well.
Right. So I think the one key thing that we do, but when we started sourcing data from different public resources, the one key thing that we realize is that the best thing to do is to take the most rawest form of the data from there. So we start with the raw FASTQ files. We would start from a state where we are not relying at all on the curation or the counts that are provided by the authors. We'll go back to the original data and what you would generate from a machine, and then process it through our standard pipelines and then make that most available to our users. So that is one thing that we try whenever that's why we are not relying too much on the processing or the availability from what's been submitted, but just do it ourselves at scale at Elucidata.
Okay. Okay, great. No, thanks for all the good questions. Data is one of those things you never underestimate how bad it could be, right? So we've all been there. It's a very tricky thing sometimes, doing data work, and you don't really ever find out the level of ugliness until you're in the middle of it and realizing, oh my gosh, what did we get into, right? So that's why data assessments are done and things like that. But with Poly and other tools, I think the future is very bright, and I suspect that there will be public data help at some point, and there as we get a handle on the data in the world, when we kick it up several notches. I know there's folks on this call that are doing a lot of work to make that happen. The world's going to be a better place.
Right.
So great job. Thanks, everybody. Thanks for your participation, and enjoy the rest of your week.
Take care. Thank you all.
All right. Bye.